Bismillah ar-Rahman ar-Rahim. Assalamu alaikum wa rahmatullahi wa barakatuh. And good morning, Malaysia. Good evening, Canada. And good afternoon, good morning, good evening, the rest of the world. Today we have our webinar series in 2024, number 39, for biotechnology, science, business, and society, with a very special guest today, Associate Professor Dr. Nehal Muhammed El-Deeb. She is from City of Scientific Research and Technology Applications, CERTA, Egypt, where we have also strong collaborations, MOU together. And she is a visiting researcher currently, an associate professor at the Faculty of the Medicine, University of Ottawa in Canada, where she is now settled in Canada. Dr. El-Deeb is accomplished associate professor at the Biopharmaceutical Bioproducts Research Departments with the Genetic Engineering and Biotechnology Research Institute, GEBRI, in Sirta, Egypt, New Burj Al Arab, Alexandria, Egypt. She is having actually and serving also as a visiting research associate for the Cellular and Molecular Medicine Department of the Faculty of Medicine in Ottawa, Canada. Dr. El-Deeb, she have extensive expertise span in the various prestigious institutions globally, and she is currently also acting head of the Biopharmaceutical Product Research Department in the GEBRI, and holding a position as a visiting professor at the Pharmaceutical and Fermentation Industry Development Programs in CERTA. Her international expertise, including research roles as a visiting researcher at the Indiana University, Bloomington in the United States, and at the Immunology Department in the Faculty of Medicine at Duke University in Duke, United States. Dr. El-Deeb, diverse backgrounds and international collaboration underscores her commitment and advancing research and innovation in biopharmaceutical and related field. I personally enjoy work with her in many publications. We have some collaborations before and still continuing. And we are very happy for having her with us today to deliver her talk about the novel drug delivery systems. Please, Dr. Nehal.
Okay, thank you so much, Dr. Hisham, for this wonderful introduction. So I can't find any word to introduce myself after this nice introduction. So I will go through my presentation directly. So today my topic will be about novel drug delivery systems. I'll give you outlines about my presentation today, like how is the drug delivery system, what's its definition, what is the conventional drug delivery system, and what is the novel drug delivery system. Also, I'll give you some examples from our research project about some of novel drug delivery systems, and also about some clinically approved targeted drug delivery systems, and then we will go through a conclusion of this presentation. Firstly, so what is the drug delivery? What is the definition of drug delivery? This means a deliver of this is a deliver or a method or a process of administering a pharmaceutical compound or active drug to achieve a therapeutic effect in human or animal. So the most common method, this is a method, not system. The most common method of delivery include, this is a non-invasive one, this is a preferred non-invasive one, a pre-oral through mouth, topical through skin, transmucosal through nasal, buccal, sublingual, vaginal, ocular, and rectal, and for sure inhalation routes. Many medications such as peptides, proteins, antibody, vaccines, and gene-based drugs, it's generally not be administered using these routes because they might be susceptible to enzyme degradation, maybe the digestive enzymes, or cannot be absorbed into the systemic circulation, maybe due to molecular size issue or some charge issue. So the protein, peptides, and antibody and gene-based drug should be delivered by only injection. So what is the conventional drug delivery system? Now, this is a system, not method. So the common conventional drug delivery system is tablets, capsules, granules, ointment, and so on. So these drug delivery systems have some limitations, like it's poor absorption from target sites, a poor bioavailability, and also high first pass metabolism. This means high first pass metabolism. This means we could lose measure concentrations of our compound or our active ingredient before the drug reaches to the rest of the body. This means through its passage from digestive system, hepatic portal system, I mean liver, before reaching to the rest of the body. So for this reason, the researchers started to develop a novel drug delivery system. So novel drug delivery system, sometimes called a controlled drug delivery system. This is like a combination of advanced technology, how we combine advanced technology and the novel dosage form or a novel material to introduce better drug potency, how to control drug release, provide greater safety, and to target a specific tissue or the desired tissue. So there are three modes of novel drug delivery system. This is three modes: targeted drug delivery system, controlled drug delivery system, and modulated drug delivery system. And today, I will only focus on targeted drug delivery system. So, what is the definition of targeted drug delivery system? Sometimes the researcher called targeted drug delivery system as a smart drug delivery, because this is a smart drug delivery, selective and effective, transfers the drug to the pre-selected target organs in certain therapeutic concentration, while restricting its access to the non-target normal cell lining in order to minimizing toxic effect and maximizing therapeutic index. But still, the delivery of drug to their site of action as a target drug delivery system is one of the major problems facing the pharmaceutical industries. But nowadays, the nanomedicine field have given a researcher a solution to overcome this problem, which plan to use a nanoparticle-mediated drug delivery in order to overcome or downfalls the conventional drug delivery disadvantage. So, I just want to summarize. We have two branches: the novel drug delivery system and the conventional drug delivery system. So, in the novel drug delivery system, we have targeted drug delivery system. In targeted drug delivery system, we have two targeted approaches. The first one is passive targeting. In passive targeting approach, we depend on the natural properties of drug carriers and the target site, both either drug carriers, the nature of drug carriers, or target site, or both. Like, for example, under certain conditions like inflammation or growing of tumors, the endothelium of blood vessels become more permeable than in healthy states, comparing with the healthy cells. And also the rapidly growing tumor, as we see here in the right side of the slide, the rapidly growing tumor recruit new vessels or engulf existing blood vessels, and these newly formed leaky vessels allow selective and accumulation much more macromolecules, which is larger than 40 kilodalton and nanosystem to tumor stroma. So this inflamed tissue or tumor cells or growing tumor cells accumulate much more nanoparticles or macromolecules comparing with the normal cells. While in active targeting, we actively or modify the carrier surface with a specific ligand complementary to certain target on the diseased cells or the target cells. So the active targeting is specifically targeted certain tissue. Okay, so there is different target organs we can target in human, like brain, lung, kidney, colon, liver, spleen, lymph nodes, and bone. So In active targeting, we could modify the surface of the carrier or drug delivery system using different macromolecules like polysaccharides, certain antibody compatible to the antigen on the tumor cells or disease cells, some protein, some peptides, some hormones, some folic acid, like it can accumulated in diseased cells. While in passive targeting, it could be depend on the particle size or the surface charge. Here is a different type of materials that could be used in the targeted drug delivery system, like polymers, nanoparticles, liposomes, outer membrane protein of virus particles, like a different nanoparticle like gold one, silica, and also the most recent materials like carbon nanotube and graphene. So I'll give you a small examples from our research project that accomplished either in Egypt or USA or even in Canada firstly about passive targeting. Here in passive targeting, there is the first example from our research project for how to passively target the colon using our microencapsulated lactic acid bacteria. So here our in vitro results confirmed that some strain of lactic acid bacteria that colonize the colon showed ability to control H. pylori infection. And H. pylori, this is the bacteria that induce gastric ulcers, gastric inflammation, and even gastric cancers, and this is the only bacteria that classified as a carcinogenic bacteria. So our in vitro result confirmed the ability of this lactic acid bacteria to kill H. pylori and control its inflammation. So we try here to microcapsulate or protect the lactic acid bacteria through its passage from digestive from stomach to its target site. This is which is the colon. So we coated lactic acid bacteria with two type of polymers. The first one is the outer one is the chitosan or chitosan, which have ability to complete solubilize in acidic conditions. So it will be dissolved in the stomach. Then the second layer will be alginate, which will swollen. This alginate have ability to swollen in the alkaline pH. So we found that the survival of this microcapsulated lactic acid bacteria, the survival rate of this bacteria, which we microencapsulated, it was significantly higher than that of the free cells after we exposed these microencapsules to simulated gastric intestinal juice, which is pH from 2, then to a pH from 6.5 to 7. Okay, so we use these microencapsules to treat induced H. pylori gastric infection rabbit model and check if this microcapsule show the ability to eradicate this animal model or not. So we found that this novel therapy with a specific dose and have certain concentrations of lactic acid bacteria showed ability to reduce H. pylori infection load in the stomach and also control the gastric inflammation due to its ability to regulate TNF-alpha expression in the gastric, in the inflamed tissue in the gastric. So this is a positive microcapsule action could achieve after receiving 24 microcapsules dosage for only 2 months. Here is another passive targeting examples from our research projects. Depending on the senses or newly synthesized nanoparticles, gold nanoparticles, using a unique approach. This is a stable one, a stable nanoparticle one to treat breast cancer cells. So as I explained before, that cancer cells show the ability to accumulate nanoparticle with a much more concentration than the normal cell due to it has the endothelium of the blood vessels at the tumor site more permeable than the healthy state. Okay, so we prepare this unique feature of a single nanocrystals gold nanoparticle with size about 0.23 nanometer, we found that the cancer cell accumulate much more gold nanoparticle inside different compartment of the cell, especially in the nuclear, in the nucleus, in its nucleus. Also, we found that the selectivity index of breast cancer cells comparing with normal cells reached 20. This means every one cells, normal cells affected by nanoparticle will be equal to 20 breast cancer cells affected by this treatment. So the ratio will be 20 cancer cells to 1 normal cells, which indicate how is this nanoparticle is very effective in treated breast cancer depending on the passive targeting approaches. So what about the active targeting approaches? We have a very interesting project to target rheumatoid arthritis. This is a kind of inflammatory induced disease. By inflammatory induced disease, when you used we used some anti-inflammatory drug. We want to load it in lipid nanocarrier and then decorate this lipid nanocarrier with hyaluronic acid in order to target the site of inflammation. Why we select hyaluronic acid? Because hyaluronic acid is the target or the ligand for CD44 receptor, which is overexpressed in rheumatic joints or overexpressed in CD44 T cells, which present in the site of inflammation. So we use this model, animal model. We induce rheumatoid arthritis in this model, and then we formulate our formulations using this anti-inflammatory drug and coated with hyaluronic acid at the outer cellular membrane of the formulation, and we start to treat this animal model using our targeted formulation for 40 days. So we found that after 14 days, we found a joint healing after treatment with hyaluronic acid coated drug, coated nanoparticles, lipid nanoparticles, and also the hyaluronic acid coated nanoparticles increased the bioavailability of drug upon its administration comparing it to the non-coated drug. So here is the summary of our in vivo studies. We found that our targeted drug delivery system increased the bioavailability of drug and also reduce TNF-alpha serum level expression and serum level, and also TNF-alpha expression at the site of inflammation. This is maybe that our targeted drug delivery system using hyaluronic acid is selectively targeted CD44 receptors that overexpressed in rheumatoid joint. Another one, we have another one, but in this time we try to treat breast cancer cells. So our target here is the breast cancer cells, and our drug is one of the chemotherapeutic agent. Most popular chemotherapeutic agent is the tamoxifen. So we want to deliver tamoxifen to the breast cancer cells. So here we formulate a nanocomposite using in the core tamoxifen and coated or decorated outside from the folate. The folate is overexpressed on the surface of breast cancer cells and accumulated and have much more accumulation rate on the cancer cell comparing with the tumors with the normal cells. Oops, I'm sorry. Yeah. So after treatment of cancer cell, we found that our formulated nanocapsules showed very promising results and accumulate inside the breast cancer cells, and we are able to decrease the IC50 value of the used formulation by 12 folds comparing with a non-targeted tamoxifen formulation. So I'll give you also some clinically approved targeted drug delivery systems that already have FDA approved and already present in the market. The first one is abrixane. This is like albumin-bounded particles from paclitaxel. Paclitaxel is the most famous chemotherapy used in the treatment of breast cancers, non-small cell lung cancer, and pancreatic cancer. So they coat the paclitaxel with albumin nanoparticles. This is a kind of protein which is accumulated by cancer cells, much more accumulated by cancer cells comparing to the normal cells. This was approved by FDA in 2005 for the treatment of metastatic breast cancer, and presented. I think it's also in a protocol in the USA, and a cancer treated protocol in USA. So the other clinically approved targeted drug delivery systems using doxorubicin here. They prepare doxorubicin in liposome. This is a kind of nano liposomes using a new technology like stealth. This is technology have developed to make a liposome evade the recognition and uptake by immune system, to protect the liposome from the immune system. This is a new liposome and have some kind of targeted particle, but there is a few information available in this product. So this product is for the treatment of patients with ovarian cancer who have recurrent infection after platinum-based chemotherapy. So the last clinically targeted approved drug delivery system is Ambisome. This is antifungal. This is an antifungal medication, but this medication used in the treatment for serious fungal infection, like like infection with leishmania, and also some serious infection with aspergillus or cryptococcus. This is here they use like a single bilayer liposomes with a double layers phospholipid, double phospholipid layer in order to target the fungal cells itself. So here is my conclusion of this presentation. Since 1990, there is a list of FDA-approved nanotechnology-based products, and also there is many clinical trials that increase nowadays by using many nanotechnology-based products like polymers, liposomes, formulations, nanoparticles, metal nanoparticles, protein nanoparticles, and so on. But there is still a regulation for nanomedicine usage depend on the safety toxicity assessment. Like we showed first, no, it's a safety and it's a toxicity, like a selectivity. It's a selectivity index, how this product will be safe and how this product will be toxic, or how this product will be effective. So thank you so much for this kind of invitation. And any question?
Okay, thank you, Dr. Nehal, for insightful presentation. We have gained valuable insights into the innovative approaches in drug delivery system.
Thank you.
Now we move on to the Q&A session. Okay. So is there any questions? As audience, please type your questions into the chat box and I will read it, and Dr. Nehal, Professor Dr. Nehal will answer it for you. Okay, I think while waiting, let me ask you a few questions of my own.
Okay.
All right, okay. The first question is, you, in your presentation just now, you mentioned a few products that have been approved for commercialization.
Yes.
So can you touch a bit on the regulatory considerations or hurdles that developers of novel drug delivery system need to be aware of during the development process?
Yes, I think the most important thing here before developing this kind of product based on nanotechnology is how to evaluate its safety and its effectively, like as I said, the selectivity index, how we could the side effects comparing with the most beneficial effects what we could have from this product. Like if we have a minor side effects, what's about its safety on the genetic mutation of normal cells that could be arise in the future while using this product or not? I think could be the core of safety usage of these nanoparticles.
All right. Okay. Yeah. Okay. Now I think I have a few questions now. So I'm going to read the questions for you. Okay. Okay, the first question is from Nur Fadila. Assalamualaikum, Doctor. What are the key challenges in designing and implementing novel drug delivery systems?
Yeah, I think the key challenges now is how to find a new material, how to find a suitable material for designing a very good or smart drug delivery system. How could we select a certain material with certain properties that could specifically select this target site or cancer cells and avoid the normal cells. I think the main challenge is the material itself.
Okay. Right. Thank you, Dr. Nehal. So I think we move on to the next question. Okay, it's from Siti Aliani. So hi, good morning, evening, morning in Malaysia, evening in Canada. Okay, I have one question. Can you explain the role of nanotechnology in advancing novel drug delivery systems?
Okay, I think I explained this in my presentation. Like, nanotechnology now is a very important in developing novel drug delivery or advanced drug delivery system in targeting certain drug either to cancer cells, that cancer cell accumulate much more nanoparticles than normal cells, or to prepare like oral lipid nanolayer to target the site of inflammation, or even using nanoparticles to control some kind of bacterial infection like silver nanoparticles. I think this is the most popular now. I think nanotechnology now is very famous as a drug delivery system. Yes.
Yes. Okay. Thank you. Moving on. Okay, from Ida Madiha. Thank you so much for sharing. How do novel drug delivery system address the issue of drug stability and degradation?
Yeah. This is a very important thing to think about it. So we are able to prepare novel gold nanoparticle or nanocrystals that could be stable over 2 years, because this is a very important very important factors that could affect the drug stability and degradation and over its half-life time or shelf time. So I think it's very important, and also depend on the material. So I'll go back to my main point. Is it depend on the material itself, how we are smart to select a certain material that have like a bioavailability, have stability, have a long time of shelf life, and something like
that. 2 years shelf life is amazing.
Yeah, we have it. We have it. Two years. Yeah, we have it. We prepare. This is a stable gold nanocrystals.
That is amazing. It's very difficult to get stability. Wow.
Yes, this is a biogenic one. This is not chemically synthesized. This is a biogenically one.
Wow.
We prepare it from polysaccharides extracted with some algal and prepare it, and we have it stable for 2 years.
Amazing. Okay, on to the next question, please. From Ifaliza. How do novel drug delivery system enhance the pharmacokinetics and pharmacodynamics of drugs?
Oh, okay. I'm not like expert in this this area, pharmacokinetics and pharmacodynamics. I'm not I'm not this person to answer this question. But I think will be also depend on the bioavailability of the drug itself. I think so.
Mhm. Right. Okay, moving on to another question from Fiz Mahmoud. Okay. Okay, so your group did a lot of drug targeting to cancer cell lines and also targeting to some diseases. Is there any application of that drug targeting? Can you target to cancer stem cell?
Yes, we published one recently, but we didn't. We still in the revision process. So I think after revision, I will share it with you. We have
Yeah, you cannot reveal.
yes.
Because there will be some issue. Yeah, of course, yes, I understand that.
Yeah.
Okay, next on is from Shahira. What ethical consideration should be taken into account when developing novel drug delivery systems?
I think this is depend on how your novel drug delivery system is safe. So nowadays we hear about the mRNA vaccination and gene therapy and nanoparticles. So I think it should be depend on its safety on cells at cellular level and then at human, maybe at healthy volunteers. So I think the first ethical consideration should be its safety.
Mhm. I agree. Yeah,
Yeah.
until we know the safety of the material, then we
Yes.
we
Yes.
would try to
Because
avoid
this is a new, yeah, because this would be a new material, a new preparation. Yeah.
Yes, I understand. Yeah, I agree with that. Okay. Any more questions? Okay. Okay, so that's the last question. Maybe I'll give. I have another one short question before we end up our session today. Okay, Dr. Nehal, how do you envision the future of drug delivery evolving with the advancements in technology and research?
I think the future of the advanced drug delivery system will have no border.
Yeah.
So we can, yeah. So we can imagine anything now. So we can see now approved nano drug delivery system based on nanomaterials. So what is the next? I don't know. I think this is the top one.
Yeah, yes, yes, yes. Okay, I think maybe that's all for today. It's already late also over there in Canada.
Yeah.
So I will end the session. So thank you
Thank you
to
so
Assistant
much.
Professor Dr. Nehal Muhammad El-Deeb for sharing your expertise and insights with us today. Your research is truly inspiring, and we appreciate your time, Canada at night, and dedication to advancing the field of drug delivery system. To our audience, thank you for your participation and especially your active participation. A lot of questions today. We hope you found today's webinar informative and engaging. And I think basically, okay, so back to Prof. Hisham. Prof. Hisham is here, so he will conclude.
Okay. Thank you, Dr. Sen.
Okay, thank you very much, Associate Professor Dr. Azila, for this excellent moderation today. And a great thanks to our guest today, and hopefully physical in Malaysia soon, Associate Professor Dr. Nehal.
Inshallah.
Even though it's a nano lecture, short time, a little bit, but very efficient.
Okay,
It's a nanoparticle. And I would like to hope, and I hope before the end of this year, we're going to see collaboration with Dr. Azila together and Dr. Nehal,
yes, for sure.
or maybe some joint publications
yes,
and
sure.
joint projects with Egypt, Canada.
Yes, for sure.
Inshallah.
Inshallah.
And at the end, I would like to express my sincere thanks for all of you and the team For the IT team, Mr. Azhari, and Mrs. Farah, and Mrs. Sabiani, who coordinate this team with us today for this meeting. And again, thank you very much, and looking to welcome you again for more and more and more collaboration. Thank you very much.
Thank you.
Salam
Thank you.
Thank you so much. Thank you.